let us see about beta oxidation of fatty acids. To give a brief introduction, this is an important energy yielding pathway by which the stored triglyceride molecules are broken down into free fatty acids, which are then oxidized to help with energy production. In the mammalian heart and liver, as much as 80% of the energy requirements can be met through beta oxidation of fatty acids. There are several steps involved in this, and in this video we are going to see in detail all the steps involved, starting from the mobilization of fatty acids from the adipocytes to the free fatty acids entering inside the mitochondrial matrix, which is where the actual beta oxidation proper takes place. So whenever there is a reduction in the blood glucose levels, or when there is an impending activity and the body requires energy, the hormones epinephrine and glucagon stimulate an enzyme called adenylyl cyclase in the adipocyte plasma membrane, and this enzyme triggers the lipid droplets to be acted upon by lipases, because the first thing that has to be done is the triglyceride molecule has to be broken down into free fatty acids. And as we know, a triglyceride molecule is composed of a glycerol backbone with 3 fatty acids attached to it. Now, the 3 types of lipases are ATGL, adipose triglyceride lipase, which first acts on the triglyceride and breaks it down into a diacyl glycerol and liberates one free fatty acid. This is then acted upon by HSL, or hormone sensitive lipase, which then breaks it down into a monoacyl glycerol and another free fatty acid is liberated. Finally, this is acted upon by MGL, or monoglyceride lipase, which breaks this down into a glycerol molecule and another free fatty acid. So now we have the free fatty acids in the adipocytes, but they have to be transported to the target cells. The target cells can be skeletal muscle, heart, renal cortex or liver. There are certain cells that cannot oxidize fatty acids. For example, the RBCs, mature red blood cells, do not have mitochondria, so they cannot oxidize fatty acids. So these free fatty acids bind to the serum albumin and they are carried to the target cells. Once they reach, they dissociate from the albumin and move inside the cell with the help of plasma membrane transporters. Once they enter the cell, there are 3 stages in beta oxidation. First is the fatty acid has to be converted to its active form, that is the acyl-CoA form. Fatty acids cannot be metabolized as such. Once they are activated, they have to be transported into the mitochondria through the carnitine shuttle, and once they enter the mitochondrial matrix, the beta oxidation proper takes place, through which acetyl-CoA molecules are produced. Starting with the activation of fatty acids, they react with coenzyme A and a molecule of ATP to form fatty acyl-CoA, and AMP is liberated with inorganic pyrophosphate, and this reaction is catalyzed by the enzyme acyl-CoA synthetases, and this activation takes place in the endoplasmic reticulum or peroxisomes, and sometimes even in the outer mitochondrial membrane. So the first step is done. Now this activated form, which is the acyl-CoA, has to be transported into the matrix. So that happens through the carnitine shuttle. There are 3 steps involved in the carnitine shuttle, the ultimate aim of which is to bring the acyl-CoA from the cytoplasm. So here we have the cytoplasm. This is the outer mitochondrial membrane. This is the inner mitochondrial membrane, and we have the matrix here. In between the outer and inner mitochondrial membrane is the intermembrane space. So the acyl-CoA has to pass through the outer membrane, through the intermembrane space, and then into the matrix. In the outer mitochondrial membrane, there are pores that are present. These are formed by proteins called porins, and it is through these pores the acyl-CoA can enter through the outer membrane into the intermembrane space. First, there is an enzyme called CPT1 or carnitine palmitoyl transferase 1. The job of this enzyme is to add a molecule of carnitine. Add a molecule of carnitine to the acyl-CoA, thus converting it into acyl-carnitine in the intermembrane space. So CPT1 adds a molecule of carnitine to the acyl-CoA and generates acyl-carnitine into the intermembrane space. In the inner mitochondrial membrane, there is an enzyme called CACT or carnitine and acyl-carnitine translocase. This enzyme transports the acyl-carnitine into the mitochondrial matrix. So now we have the acyl-carnitine molecule in the matrix. But acyl-CoA is the only form that can be metabolized, right? That's the active form, but we have an acyl-carnitine here. So we have to add a coenzyme A molecule here, which is done by the enzyme CPT2 or carnitine palmitoyl transferase 2. So this adds a molecule of coenzyme A here. Thus, it is converted back to acyl-CoA. So we have acyl-CoA in the mitochondrial matrix. Now this carnitine is also liberated. So the carnitine is transported back into the intermembrane space by CACT, carnitine acyl-carnitine translocase. That's why it is named as both carnitine and acyl-carnitine translocase, because first it transports acyl-carnitine from here to here, then it also transports carnitine from the matrix to the intermembrane space. This carnitine can later on be used by CPT1 for any other acyl-CoA molecule that is about to enter. So now we have the acyl-CoA in the matrix, and the acyl-CoA starts undergoing beta oxidation proper. Also, fatty acids with carbon chain length of 12 carbons or less can enter the mitochondria without the help of membrane transporters. But those with chain lengths of 14 carbon atoms or more have to enter the mitochondria by undergoing the 3 enzymatic processes in the carnitine shuttle. So let us see in detail about beta oxidation of fatty acids in the next part.